All right. Continuing along today with the discussion of phylogenetic systematics, also known as cladistics. We're going to focus on the applications of cladistic analysis today. So how is cladistics used in contemporary biology? It's used all the time is the short answer. It's fundamental and central now to analyses in ecology, evolutionary biology, epidemiology. A bunch of ologies are relying on cladistics for insights into biological systems. And I'll go through some examples for the most part during this lecture, finishing with some things from last time. We will, at the end of this lecture, be fully caught up, I think, and be able to move forward on Friday with the fossil record and deep time. So we will do those things today. Just on this first slide, you'll see this image of our island hopping model. Remember, this is a model I've repeated a couple of times. It started with Darwin. You can think of an archipelago like the Galapagos as a setting whereby a species from the mainland may get to the first island, jump to a second, evolve via microevolutionary forces into something else, colonize a new island, evolve there, and pretty soon you have this overlapping set of species that has this unique history. But if you arrive there today on a ship and find all of these creatures, you're not able to watch this history. You have to reconstruct it. And one of the ways of reconstructing that history now is with a cladogram. You can use the cladogram to infer past biogeographical movements of these organisms. So take a close look at that cladogram on your time, its topology, the implied relationships, and think about the evolutionary history and a narrative, an evolutionary narrative you might be able to tell from an analysis of that cladogram. That cladogram based on morphology or genetics that's entirely independent in the data that go into the construction of that cladogram from this geological history and these patterns of movement. Monophyly is stressed by cladists in their analysis in relation to the appropriate naming of formal biological groups. A group like the primates is a monophyletic group. Linnaeus set it up, primates, us, monkeys, apes, and friends, and it has held together through the centuries of analysis in dissecting, literally, the group and determining relationships. The primates remain a group with, as we now understand, a shared evolutionary history from a common ancestor, sharing features unique to this group, and importantly from the cladistic standpoint, a common ancestor that's exclusive to this group. It's monophyletic. The lemurs and lorises are included with the some lesser-known primates and the monkeys and apes. These are the apes, us sitting there on the far right studying or something like that, with chimps, gorillas, orangutans, and the lesser apes, the gibbons. And there are a couple of species named for that on that chimpanzee line or the gorilla line or the orangutan line. And so not all of the diversity of the apes is shown there, but that's fine. We simplify it in the cladogram because our purpose right now is not that type of resolution. It's a broader scale. So the apes, a monophyletic group within the monophyletic primates. The monkeys. Now notice the monkeys do not, as a group, they're in quotes, first of all, monkeys. And that's a reference that we use to denote a group that's not monophyletic, not a true clade, but maybe a group that's convenient to refer to, because most primatologists know what you're talking about when you say a monkey, even though it doesn't refer to a natural group. Because look, all of the descendants of this group are not included. Well, so you have your common ancestor and not all of the descendants in a grouping that you might call monkeys. Thus, the group is paraphyletic, right, as opposed to monophyletic. But everyone knows what you mean. So it's convenient. It's still convenient to biologists to refer to monkeys. So who is that? Yes. That's Curious George, right? But the man with the yellow hat always called it a monkey, yes? Always referred to George as a little monkey. Now, George has no tail. The apes lack tails. It's a synapomorphy of the apes. It's something that helps to diagnose the ape clade is taillessness. All the monkeys have tails. And some of the monkeys, the New World monkeys, have prehensile tails. All right. George must not be a monkey, right? Lacking a major feature of the monkeys. So do we quarrel with the author of Curious George? No, I don't think so. You can if you want. But that's an uphill battle if you start to chase monophyly as a necessity for all of our terms that we use around organisms and in everyday English. If you're a stickler that way and say, you can't call it a monkey, you must call George an ape, then you're going to have a lot of debates in your life. And a better option may be just to use your air quotes when you say the term, or just let it slide and consider this colloquial English and not formal scientific terminology. Both are good options for steering your way through the morass of language around organisms. Let's look at another example that's really important in the modern context. And it's fascinating, a place where Berkeley led the discussion, just like Berkeley led in the phylogenetic systematics revolution. This is a discussion that Berkeley has led on. The nature of the reptiles. Most textbooks will establish, at least up until a generation ago, among the tetrapods, amphibians, birds, reptiles, and mammals, right? And everyone knows what the reptiles are. They're the scaly creatures that tend to slither along on their belly, low to the ground, and sometimes have a forked tongue, right? Lizards, snakes are reptiles, turtles, reptiles. It's an old term, and everyone knows what it means. But there's a major problem with this term, and it's a problem on the scale, such a scale that you might consider even abandoning this term in formal usage completely. It still hangs on in this department, but watch this space, because it'll be interesting to see what we're doing with the term reptiles in the future, and why. Why is this a problem? Does anyone have an inkling or know something about this subject? Why is the term reptiles a problem? Yeah. Didn't birds evolve from them? Yes. That's become quite clear on a whole bunch of analyses, including molecular analyses and analyses of fossils, that the birds, right here, are nested high within a big group of reptiles. Specifically, birds evolved from types of dinosaurs, the theropod dinosaurs, the group that T. rex belongs to. Those dinosaurs have similarities in their skeletons that are extremely close to the features seen in the skeletons of birds. There are dinosaurs that are found with feathers, bridging the gap nicely between these dinosaurian groups and the birds themselves. In this sense, the birds are reptiles. Indeed, the birds are dinosaurs by our terminology, by our formal terminology. Birds are literally feathered dinosaurs. And so the whole concept of an extinction of dinosaurs, you have to sort of pause and think about, not all the dinosaurs went extinct. We see them every day. They're feathered and flying all around our skies. A big group of dinosaurs went extinct at the end of the Cretaceous, 65.5 million years ago, but many survived in the form of these feathered types. So the reptiles is seriously paraphyletic if you're excluding the birds. So one of the nice solutions is to keep using the term reptiles, but guess what? Birds are in the group. If you say reptiles, you should intend birds to be included if you want to drop the air quotes on reptiles. Yes, please, in the back. Yes, turtles are and always have been problematic as to their position in a cladogram. Turtles have this funny shell, right, with the ribs on the inside, and they're all flipped around, and the skeleton's all crazy, and they show up in the fossil record just as crazy as they are now, and they're hard to interpret and place on the cladogram. So the dotted line indicates ambiguity of its position in the evolutionary phylogeny. They bounce around. Every couple analyses, you'll see the turtles moving around, not entirely clear where they belong. It's like bats in the mammal world. Bats are so highly modified for flying. The first fossil bats already have those hand wings. It's hard to decipher where they came from in the phylogeny of mammals because they're so overprinted by changes that they're hard to read. A species concept can be based on phylogenetic principles. A species being recognized as the smallest possible monophyletic group that's diagnosable from other such groups. And you determine what species exist through phylogenetic analysis. A species has to share a unique common ancestor to the exclusion of other species. And it has to be distinctive by some measures, morphological or genetic or whatever they may be. And so this whole ring species might only be diagnosable as a single branch on a phylogeny like this as a result of sharing a single common ancestor that came down from Oregon and formed these different types in California. The ring species example of Encetina might just be a branch tip on a phylogeny in an analysis and thus would be considered a single species by these criteria. So the phylogenetic species concept solves some of the problems we saw with the biological species concept or a morphological species concept or any of those other dozen or more species concepts for a lot of people. So this is the phylogenetic systematics revolution really expanding its reach into our taxonomic recognition of these categories. Remembering that species is just one of these ranks in the whole Linnaean hierarchy. And a strict cladist will just want to talk about phylogenies like this, branch tips and phylogenies, and naming these organisms on the basis of their distinctiveness and their distinct evolutionary histories. A strict cladist won't care. Well, is that a species? I don't care. It doesn't matter what rank it is. It's a tip on my phylogeny. Sure, let's call it a species if you really need to define this rank. And we do. One of the major reasons we do is for political reasons. There's a whole political structure based on those categories. There's an Endangered Species Act. Do you think you want to get into a debate with politicians about the nature of species if you're concerned about a particular species and trying to argue against the extinction of a species? Do you want to start stepping backward and arguing species concepts? Biologists can't even come to terms on this, let alone biologists and politicians. So the cladist will say, in the scientific context, don't worry about the rank. It doesn't matter. But in practice, we need to worry about the rank. It's a critical component of our social discussions about the biological world. The construction of cladograms is something you've started working on in lab if you've had lab this week, and you'll continue next week. You'll see all of this material, so I'll just go over it quickly here just to show you the terms involved. You'll be building character matrices with taxa lined up against these characters, and you'll be scoring characters according to their presence or absence in the taxa of interest. The taxa of interest that you're interested in studying, well, that's called your in-group, right? You identify the the organisms you're interested in investigating. That's your in-group. You choose an out-group that's primitive in relation to your in-group and lacks the characters that your in-group exhibits. So in this case, the lancelet lacks all of these features that are exhibited by some members of your in-group. And that's necessary to polarize your characters, to understand the changes that happened over evolutionary time. Again, you'll be getting all of this in lab. And then you'll use those characters to build a cladogram, to generate the topology of a cladogram. And this is a cladogram built just on that previous character matrix. And you'll use computers to assist you in choosing the best cladogram. The principle of parsimony is the one that's relied upon in lab for the selection of the best cladogram given the available data. The principle of parsimony goes back to this guy, William of Ockham. It's also known as Occam's razor. If you've had intro philosophy, you might have been introduced to Occam's razor. It's the principle of parsimony. And it's basically common sense. Don't make any assumptions. Don't make any more assumptions than necessary when investigating something. You do this when you lose something. You lose your phone. You don't go look at the store if you know that you just made a phone call from your house after you return from the store. You look in your house first if you're using common sense. If you're not making any more assumptions than necessary, such as someone stole my phone from my house and then lost it at the store where I just shopped. That's a gross assumption in this context, totally unnecessary, and will lead you astray about the true history of the fate of your phone. It's really similar to what we're doing in cladistic analysis. You're choosing the simplest possible narrative for the structure of evolutionary relationships, of the taxa of interest. And according to that character matrix, we had this cladogram built from the available data. You could have drawn the cladogram differently. You could have drawn it with a topology like this. But when you map those characters onto the cladogram as synapomorphies, you'll see that this construction, this cladogram, requires 6 evolutionary steps, including the evolution of an amniotic egg twice, as opposed to just once. More steps are necessary here to describe this evolutionary process than here. And by the principle of parsimony, one major criterion that cladists use to choose among alternative cladograms, this would be the preferred cladogram. It's simpler. There are fewer assumptions, fewer evolutionary steps necessary to explain the topology. Those were morphological features, right? Whether you have an amniotic egg, whether you have 4 legs, or what have you, whether you have feathers. Genetics have overtaken morphological analyses in their prominence today, molecular analyses at the genetic level. The number of base pairs available for analysis is so many that you just have a great many more characters available to you in the DNA. And you'll practice with plastic beads simulating base pairs in lab and work some of these phenomena out just like a molecular biologist would. And yet the morphological information isn't gone. Even behavioral information that can only be gleaned from studies in the wild, those are still really important to scientists, although they've faded in importance. So the genetics and molecular revolution has really changed our approach, but there's still a lot of fundamental and interesting work done in the morphological sciences. And it's in some ways a shame when you lose so many practitioners to the other discipline because of the insights it's giving you. You're losing insights by moving away from some of the more traditional approaches. But certainly it's the case that the molecules have revolutionized our understanding. They help shed light on organisms that may be impossible to study in the wild because they've gone extinct, because they're in too remote a place, because they're in a place where local conflicts don't allow biologists to enter. Any number of reasons why organisms can't be studied in the wild or exist as too few specimens in a museum. And we need to use the DNA to study them. Birds are undergoing a major reconfiguration of their relationships. Bird relationships were fairly stable over many decades until molecular analyses were applied to them. And that tree's been shaken hard a couple of times. This was a co-authored study by Berkeley biologists and others from around the world, just the first in the last several years to take on these global analyses of birds and really changing our understanding of bird phylogeny. Really fantastic if you're an ornithologist. Really exciting times. Frustrating times for the authors of bird guides because they need to rewrite their guides and reorient the pages to accommodate changes in phylogenetics, and that's frustrating for the amateurs who aren't involved in the studies themselves. Molecular analysis can shed light on organisms that, like bats and turtles, that just are so different, have changed so much that their relationships are poorly understood. One of the biggest flowers in the world are these flowers of Indonesia that rely on insects for pollinating them. These are flowers that stink and attract flies in particular to their rotting odor and are pollinated that way. The floral parts are so exaggerated, the vegetative parts so reduced that it was always hard to figure out who they're related to. But the molecules are in there. The DNA is in there, and it hasn't changed in these wild ways like the reproductive parts have. So just the application of molecular techniques helps to position this group of flowers in a larger clade with clarity. These flowers that grow in different areas, all with similarly gigantic floral parts, prove to be closely related to one another according to the molecular analysis. The molecular analysis which is independent of this morphological construction, showing them to be closely related to one another in a larger clade, and you have robust support for that position in the phylogeny. Great use for molecules on organisms that have changed so much, or in comparing organisms that are just so different, like us and fungi and plants. So humans and fungi are hard to compare because of such significant differences in body plan, cell structure, physiology. What morphological features do you choose to put us even into the same group as mushrooms and a tree? Genetic data. We share the same genetic code. We share so much genetically. And when you do that, you see that humans fall into a clade with the fungi to the exclusion of the angiosperms. So when you're eating your salad, you're more closely related to the mushrooms in your salad than the lettuce in your salad, if you want to think of it that way. And we know that from genetic analysis, not from morphological analysis. The cladograms we've been looking at until now, the lengths of the branches didn't have much meaning. The topology, the branching structure, had great meaning in determining who's more closely related to whom, who evolved from a common ancestor. But the branch lengths weren't significant. They didn't imply anything except the relative timing of divergence. Other types of cladogram can be built, call them phylograms, where relative branch length signals genetic or morphological distance or the time of divergence, or both. There are tools available to infer time of divergence in deep time of lineages based on molecular differences. Using a molecular clock and fossil calibration in combination, one can determine the divergence dates of lineages like these on a cladogram. Now, these are the animals of the world. These are animals that have had their whole genomes sequenced. Major groups have had their whole genome sequenced in the major groups of animals, and a phylogeny like this can be built showing a divergence of the tetrapods, according to this analysis, around 450 million years ago. Or a divergence of the animals themselves 1.2 billion years ago. And of course, those are open for debate. Interesting analyses can be done on this. The number of genes in these organisms, the total number of genes that they express, increases with the tetrapods. The number of Hox genes, if you've been introduced to Hox genes, these guys have just one set of Hox genes. These have 4. And so there was a doubling, and then a doubling again of Hox gene clusters, it seems, in evolution. But the point here is with regard to the branch lengths. The length of the branch is on an absolute time scale here. So you could literally take your ruler and measure the lengths of these branches to infer something about the time of divergence according to this axis. Always and only just a hypothesis, though, and open for re-examination and modification. But maybe it's our best available hypothesis. And that's the nature of science. This is the scientific walk, right? It's our best one going. Let's use it. Let's assume it for other activities. But we haven't proved anything. You don't stop there. It never stops. If you want a job where you finish and it's over and you sit down and done, science isn't it. Science just keeps going, and it's brutal in the trampling of the older analysis. But that doesn't mean it's not to be celebrated for the accomplishment here. It's just provisional, subject to revision. So some more examples in how we use cladograms. And this will just help to get you familiar with the terms, help you become fluent in the interpretation of these cladograms. If you go on safari in Africa, you'll be told by your safari guide, when you're out looking at the elephants, you'll be asked, who is the elephant's closest relative on Earth? And they'll amaze you by telling you that the closest relative of elephants on Earth, or it will be in your field guide, are these little guys, hyraxes, which are just the cutest little things. And you don't have to be a good photographer. I took this picture. You don't have to be a good photographer because they'll just sit there and stare at you for 15 minutes. And then 15 minutes later, they'll move their head like that. And then 15 minutes later, they'll swing their head like this. And they're just photogenic as can be, standing there on the rocks. And then they'll bound around, and they're very active, but they'll stay still like that for long periods. Great things. They're called hyraxes or a coney, big like a big bunny, round-bodied like this, bounding on the rocks or even in the trees. The closest relative of elephants, it is said. Sort of, kind of. I mean, if you look at a cladogram with the hyraxes and the various elephants that have existed, including the mammoths and mastodons, and then the living ones down here, you see that they're all in one big group, one big monophyletic group with the elephants, but the manatees and the dugongs are stuck in there as a branch. And you've got fully 40 million years since the shared common ancestor. So the closest living relative, manatees and dugongs, we must talk about those. Close? 40 million, is that close? Since they shared a common ancestor? But don't quarrel, just let your safari guide talk. Another great use of a cladogram in the study of the evolution of wings in stick insects. Just to give you an example of how this type of mapping work is done in an analysis of something like flying in this insect, once you have a cladogram, you've built your cladogram from molecular data, and then you take the morphological data and map it onto your cladogram, which is developed through independent means, molecular means. And when you look at something like the gaining of wings in this phylogeny, these blue stars, you see that wings seem to have existed in the basal lineages. The blue lineages are the winged lineages. And wingedness looks like it's basal to this whole group. It's the primitive condition in this in this clade. All of these guys lost their wings, so that these lineages that have wings must have reacquired them, because the inference is that their ancestors didn't have them, based on a parsimony analysis in the mapping of this morphological structure. So the winged stick insects, which you could easily be fooled in an analysis that didn't use molecules to thinking that all the winged stick insects, they must be one group, and all the wingless ones in another group. Let's name the winged clade such and such, and the wingless clade something else, and keep them separate in our museum drawers. Well, this screws all that up and makes it a lot more interesting, with the notion that wingedness was primitive, it was lost, and then regained, reacquired multiple times. It's not the most parsimonious explanation that you could devise for wings. The most parsimonious would be to think they were primitive and lost once, or gained only once. Well, the natural process doesn't always occur in the simplest possible way. There are multiple routes possible. And so parsimony, naively applied, can lead you quite astray. Or in analyses of biogeography, cladograms are often fascinating. Biogeography, the analysis of the spatial distribution of organisms, in this case on a planetary scale, across continents, with a phylogeny of bats, a total evidence phylogeny. Total evidence including, usually as a reference to the inclusion of multiple types of data, not just molecular data used to construct this, but morphological data, and including fossils. So it's all the evidence available combined into a common analysis. To include the fossils in here, you usually need morphological information because the DNA isn't available from the fossils. It's very rare still to extract successfully ancient DNA from fossils. That's sort of a popular myth that scientists are getting ancient DNA from all these old organisms. More and more success is being had in that regard, but it's still quite limited. So the bats in this phylogeny that are extinct are placed based on morphology. And when you have a cladogram like that built from a total evidence package and then map biogeography onto it according to where these bats live, you see some interesting patterns. The basal lineages are North American in yellow. So all these lineages that form the outgroups to this other clade are in North America. What does that imply, perhaps, in your narrative of evolution of bats? What's the implication? Anybody? Can you learn anything from this colored cladogram about the evolution of bats 40, 50 million years ago? Where did it happen?
The common ancestor came from North America.
Yes, that the common ancestor of these bats was most likely to have existed in North America. You can infer that from the nature of the branching of the cladogram with a knowledge of where these bats are now, unless there's been some shuffling and a returning, a less parsimonious explanation is available. But no, it looks like the early bats represented by these lineages were in North America. Maybe bats, the crazy bat with the flies with its hands. Where did that start? Maybe North America. And then maybe spreading to Europe, if you want to interpret this strictly. And then these bats spreading to Europe, so across the northern hemisphere, and that's a major pattern in mammals. That mammals are holarctic, across the northern hemisphere or just in the southern hemisphere. Maybe they spread across the northern hemisphere into Europe and Asia, right? In these two groups, light blue and purple, Europe and Asia. And then into the tropics, in these highly derived clades that entered the tropics only later. It's a believable story, quite believable. There's some interesting there's been much more since this, 2005, much more on bats since then. showing the timing of the evolution of bats by some registers being correlated with the timing of the evolution of insects that they might have fed upon. But neat stories available based on cladograms. Here's a story, if ever there was a story. And I don't mean to tell you that this is true, just it's an example of how a cladogram can be used in an analysis. And this is an archaeological type analysis in the study of human clothing. When did humans in an evolutionary context start wearing clothes? You know, if you think about a naked history, when did clothes start to be put on and worn regularly? Again, take this for what it's worth, but shows you the possibilities of an analysis like this. What these investigators did was to study lice. And if you know something about lice, and if you don't, when you're a parent, you'll know something about lice. If you're not a parent already, my gosh, it's common, particularly in California. Californians, I don't know, I grew up on the East Coast, and we didn't have, at least back then, I don't know if it's a time thing or a geography difference, but it seems that our climate here is, especially in the summertime, late summer, early fall, kids are lousy little things around here. And there's a couple different kinds of lice. There's lice of the hair, and there's lice of the body. And they're considered different subspecies or different categories of lice. There's the type that lives on the scalp, and there's the type that lives on other parts of the body. And these are, they're mildly annoying, sometimes not even noticeable until they get super, super infested. And they rely on human products. They rely on human products for their life, human-generated food. So when you look at a cladogram of lice, based on the molecules of the lice, the genetic analysis of the lice, you see that the basal lineages of lice are in the Old World, Africa and Europe, and they're hair lice. It's only hair lice at the base of the phylogeny here from the Old World. As you move up in the phylogeny, you start to pick up your body lice all over the world. The implication is that these lice evolved to associate with humans in the Old World long ago, based on a clock, molecular clock here, 500,000 years ago, this association may have formed. Around 70,000 years ago, you have the first lineages of hair lice branching off. Body lice. The notion is that the body lice only could survive when people started wearing clothes. So the archaeologists make the leap in their narrative to the evolution of clothing having occurred sometime around 70,000 through an analysis of lice DNA and the construction of a cladogram. Now that might be total hogwash, and probably is, but it shows you the possible types of analysis that can be done. All right, let's take a television break.
A tranquil pool in Argentina, and in it, a female, broad-snouted caiman. She laid her eggs in a pile of vegetation close to the water almost 3 months ago. Now, sounds are coming from it. The eggs are beginning to hatch. Even while the eggs are still within the nest, their mother can hear them from some way away. Back on the nest, she listens intently. Then, very gently, she starts to take it apart. She can't know exactly where each of her babies is and stops every few seconds to listen. At last, the young are free. But she doesn't abandon them. She is going to take them down to the pool that she's selected as their nursery.
All right. There's your very sensitive croc mama. Not known for this type of behavior, but it's typical of crocodilians, this parental care behavior. The mother takes them down to the water, comes back and takes them by the mouthful in this case. There's predators around, the hawks are around, the monitor lizards might be around, lots of things wanting to eat the babies along the way, and then even once they get in the water, they're highly vulnerable to fishes and other crocodilians. So the mother's taking care of them, protecting them, first letting them out of the nest, and then protecting them. It's parental care, right, on the part of this reptile. Not only that, but they're making sounds, right? They're calling. The little babies are calling, and the adults are capable of vocalizations. So they're capable of making sounds, and they demonstrate parental care based on the defending of the nest and the care of the young in crocodilians. Where are the crocodilians in a phylogeny? They're in a phylogeny of reptiles like this. Up here, having branched from a common ancestor, shared with lizards and snakes, they form a clade, the crocodilians do, with the dinosaurs, including the birds. Right? The birds are all over the place today. And we, of course, know that they make nests and defend their nests and raise their young. But we don't know as much about these guys, these big flightless dinosaurs that went extinct so long ago. Can we make inferences about these guys based on a cladogram and studies of behavior of living members of such a cladogram? These are the extant members of this cladogram. Extant as opposed to extinct. Extant, still alive today. Members of these clades are. Members of these clades are completely extinct and then extant. What might you be able to say about the behavior of these guys from a cladogram like this, given the stuff you know up to this point? Can anyone make an inference? It's challenging, but I know someone can. Yeah. So if we know that crocodilians, what he said was they probably have vocalizations. Good. I hope some of you were on that line of thinking. If we know that the crocodilians are exhibiting parental care and vocalizing, and we know that the birds are exhibiting parental care and vocalizing, is it safe to assume that about the common ancestor back here? More or less. Unless you're going to have a less parsimonious interpretation of it having evolved twice. Why not just have it evolve once, here, and infer that that was the case for these guys? It wasn't always the inference about these guys that they would have been doing that. To the extent that when one of these early spectacular dinosaurs was found on a major expedition, one of the expeditions that inspired the whole Indiana Jones thing, this expedition of this young guy out of New York to Asia, to Mongolia, they found this collection of bones and eggs. And this creature was given the name Oviraptor. Indeed, Oviraptor Philoceratops. The egg thief that loves ceratopsian eggs, because it was thought to be, and here's the hind limb and a forelimb, if you can see the skeleton in there, and then these eggs. It was thought to be a dinosaur that was raiding the nest of another dinosaur. It was the egg thief that ate the eggs of the ceratopsian dinosaur. But in the modern light, and with new discoveries, and an analysis of the little skeletons that are occurring inside these eggs, you can identify these eggs as belonging to the same species as the adult form on the same structure. Of course, it's a nest. It's the mother that's on the nest that's being found, covered by a volcanic ash explosion or whatever secured that individual on its nest in the fossil record. So depictions like these seem quite fair. And more nests have been found with a little ring structure of eggs positioned like this, where the mother can sit inside and keep the whole thing warm and rotate during the night, maybe. So we can make inferences through an extant phylogenetic bracket by bracketing ancient lineages based on extant forms that we know something about. So you can have all of these pretty pictures and more inferences like color pattern that are possible based on the extant phylogenetic bracket. Not entirely whimsical or fantastical reconstructions like those. All right. Enjoy the rest of your week. I'll see you on Friday.